all of my working life has been spent working on the human MHC. And so I've seen it go from very early stages of molecular definition through to today. And I'm going to indulge myself a little bit at the beginning of the talk and to show you that trajectory, because I think that when we started, we were getting data that we couldn't do very much with. And the more we worked on this system, the more we get out of it. But you have to be persistent and wait for it to break open. So we've got ourselves involved in 3 different genetic complexes that are involved in regulating T cells and NK cells. And there's the HLA complex on chromosome 6, which provides ligands for NK cell receptors as well as T cell receptors. There's the natural killer complex on human chromosome 12, which encodes lectin-like receptors that recognize some of the HLA class 1 molecules, notably HLA-E. And then you have the leukocyte receptor complex on chromosome 19 that encodes these KIR receptors, which interact with the polymorphic HLA-A, B, and C. And so the question that I asked when I first went into Jack Strominger's lab in 1972, where he was just starting to try and study MHC class 1 polymorphism, my main interest was in what the nature of the diversity was. And I'm just going to go through some of the papers that there were at that time. And this was really the first paper. And this was based on work that was done at the Victoria Hospital in East Grinstead in Sussex, and where it was found that one way to get hold of HLA, which is a membrane protein, and nobody knew how to extract membrane proteins, was to take a tip from Rodney Porter and do a very, very harsh papain digestion of either cells or membranes. And then you could get a soluble fraction that would interact with the alloantibodies that were being discovered by clinicians in patients who'd had lots of blood transfusions or women who'd had lots of babies. And so you could get this digestion, and it would release soluble HLA A and B, and then with relatively simple column chromatography, you could actually separate the A from the B. And when my first few months that I was in the Strominger lab, this paper was being written. And this was when SDS gels were just starting to be used. And so they just ran a gel, and they found that, in fact, there were 2 peptide fragments. And then I went away on holiday and then missed what was probably the most exciting thing, which is that these are the 2 fragments. But there's this protein called beta-2 microglobulin, which is often found in the urine of people with kidney disease, particularly in Sweden, where there were people working in a cadmium factory and who had very bad kidney disease, where they would have a lot of this protein. And some sequencing had been done on that. And this fellow, Pulik here, had made a career out of a rabbit anti-beta-2 antiserum. He'd never tried to see if it would bind to a cell. But having seen this paper, he then did that and found that the anti-beta-2 bound to cells. And so then it became clear that the small subunit was already known. And so then the analysis could turn to the heavy chain. And this is where I started to come into this. And I got my first paper for sequencing 1 amino acid. And the question was that having done this harsh papain digestion, there was the concern that you would have a lot of frayed ends and that you wouldn't be able to sequence this protein because it would be starting at a lot of different places in the sequence. And so what I did was to do standard N-terminal sequencing on the molecule and found that I only got 2 amino acids. There was the isoleucine, which was known to be associated with the beta-2, and then there was glycine. But that was another problem because the Harvard Biolabs, where I was working, was one of the filthiest places on earth. And everybody was using glycine buffers. So everything was contaminated with glycine. So coming up with glycine as an answer wasn't a very good answer. And so I had to go through rigorous approaches to do the experiments away from all of my colleagues that were slopping glycine everywhere. And so we finally managed to do that. And we got this from 3 different types of HLA class 1 antigens, so there was a consistent result. So that was 1 amino acid. And so the next paper we did, we got 25 amino acids. And this was mainly the work of Cox de Horst, and looking at A2 and B7, and there was 1 position of polymorphism. So this was how we got the first position of polymorphism. So you get a paper for the first amino acid and a paper for the first polymorphism. And this went on through a few other papers and sort of ended at this point where a number of other investigators were now involved on the project. And the whole sequence of B7 was determined, and that was lucky because there was a very fortunate acid cleavage fragment that allowed it to be divided into 2, which we couldn't do with A2. But there was about 150 residues of A2, so that we could look at we could do some comparative work, and there were lots of differences. So this involved maybe about 10 graduate student years and about 15 postdoc years. So in 1985, Bev Kohler, who was now working with Harry Orr, who was a PhD student, she was able to clone the gene for HLA-A2. And with 2 or 3 graduate student years, she did a lot more than what we had done in the previous decade. So this was how the field began to ramp up. And so this hailed the demise of protein-based sequencing, which everybody thought would go completely extinct, but then additional and new applications of that were discovered. And her work directly led to the acquisition of sequences for all the common HLA class 1 alleles during the period from 85 to 95. And this would be on the order of about 50 or 60 alleles that would be the common ones you'd find in the Anthony Nolan bone marrow transplant registry in London, or a typical European or North American population of European origin. During this period, bone marrow transplantation went from being a very experimental and not very good procedure to becoming a standard practice. And unlike kidney and heart transplantation, there's an unlimited number of possible donors so that you can make much better HLA matches when you're dealing with bone marrow transplantation than kidney. And it was clear that if you had a donor and a recipient who were HLA identical, you got much better expectancy of life, less relapse, and it was a great improvement. So with that clinical experience, there was a drive coming from the clinical arena to make HLA typing better than it was, because they were still using the serology that had been built up by people in the 1960s. And it was absolutely clear from the other sequencing studies that it was imprecise and was at low resolution. And so they started to apply DNA-based probes for doing typing around 1996, and then they started to do sequence-based typing starting in about 2006. And with each of these advances in HLA typing, people like me got more information about the sequencing, patients got better, and so there was a synergy that developed between the clinical side of the field and the genetic side of the field. And what I would suggest is that the same thing might happen between the people working with animals and the people studying animals. So this is a graph showing you the number of different alleles that have been defined over this period of time from about 1988 to the present day. And so this is where DNA-based typing started for BMT. And you can see there's a slight inflection in the curve. If you go a little bit further up, you get sequence-based HLA typing for BMT. And then it went really into exponential growth. And this is where the sequence-based typing was first initiated. And now there is a big sequence-based typing machine in Germany called DKMS, and they're routinely typing millions of people. I mean, there's probably been about 15 million people that have been typed. So that's how we got the data. And having started from 1 amino acid, we now have thousands of full allelic sequences for the exons 2 and 3, which are where all the binding site of the HLA molecule is. And so we started an analysis, and this is with Steve Marsh and James Robinson at the Anthony Lane Centre at the Royal Free. And we started that in about 2009. And it took us almost 10 years to do the analysis. Now the analysis became the rate-limiting step. And that's because we had so many sequences that James had to write all these programs for analyzing them, and he ended up having to write a whole suite of programs. And then by the time we did the first analysis and submitted it to a journal, there were another 5,000 alleles, and the referee said, well, this isn't a good analysis, you've got to do all the ones. And so then we had to go and do all that again. And so this is the title we finally came up with, which was Distinguishing Functional Polymorphism from Random Variation in the Sequences of Over 10,000 A, B, and C alleles. And it was in PLOS Genetics and just in June of this year. And so these are the players. James did the donkey work. Libby in my lab did a lot of the analysis and the biostatistics. Steve orchestrated the whole thing. He's in charge of the worldwide database of HLA sequences. Sue Young Yang and Nezzy Sereb were postdocs at Sloan Kettering Hospital in New York, who then set up a sequencing company and were the first people to really do serious numbers of typings by sequence-based typing. And the analytical methods that James developed are applicable to any large groups of sequences. And in the paper, he tells you how to get all these methods. And so you can do this analysis on any favorite gene or any favorite species. So I'm just going to give you some examples of the data. So here he's taken all these 10,000 sequences and looked at them by pairwise comparisons. These are the HLA-As, the HLA-Bs, the HLA-Cs, and then D is a mixture of all three. And what you see that you get these areas of color, such as down here and down here, and those correspond to the old serological antigens. So there's a good correlation. It's just that the resolution of the serology isn't as good. He's got this other program at looking how different alleles relate to each other. And what we found, not surprisingly, is that most of these alleles are actually rather rare, and they differ from a common allele by just one nucleotide substitution, so they're SNP alleles. And so this is a graph of HLA-A2, which is the most frequent serological antigen in the English population, the European population. And so all of this lot, these are all SNPs, just one nucleotide difference. And these are other collections here, and the way that James sort of invented this is that you can only join things if they have a SNP in common. And so some of these ones that are out here by themselves differ by one SNP that hasn't been found anywhere else. And so something went on where it got that SNP, and the intermediates are no longer with us. So from these alleles, we found there are basically three categories. They're the SNP alleles, which I've mentioned, which are over here. This is just a tabulation. You've got recombinants, and recombination is particularly common, particularly in small indigenous populations that have gone through strong selection, where you select for recombinants, such as in the South American Indians or in Australian Aboriginal populations. And then there's the other ones, some other ones, the sort of deletions and things like that. And then you've got some what we call core alleles, which are alleles that are so different that they can't be derived simply from any other allele. So let's have a look at the sequences. So what we found is that for A, B, and C, and we're looking at 182 different positions in the protein sequence, there are at least 3 alternative amino acids at every position. And the number of alternatives goes up to 14. So you've got lots and lots of diversity in A and the B and the C. This is just looking at the number of differences. If you then do the following analysis, you want to try and get rid of polymorphism that's really rare. So you look at not the most common position, the most common amino acid at a position. You look at the one that's the second most common. And if the second one is also there at some frequency, then you know you've got a real polymorphism. So you've got at least 2 amino acids that are at significant frequency. And that changes the pattern completely. And it's a much cleaner pattern. And this is exactly the same pattern that you get if you're a crystallographer who's looking at crystal structures and asking where are these proteins interacting with peptides and receptors. And it's an absolutely perfect correlation. And so the polymorphism is tracking exactly with what and these are 2 independent assessments. And if you do selection analysis, which you can, given all these sequences, that correlates as well. So it's really a beautiful correlation. And it's sort of the question that I started off asking so many years ago is now completely solved with this type of analysis. Here again with HLA-B, you see the same thing in HLA-C. You can do the same analysis with the nucleotides, and you again get these rather striking patterns where these nucleotides are in the codons that we were looking at in the previous. And here you can begin to see polymorphisms that are functionally important, that are specific to one gene or another. And there's quite a lot of them that, as you might expect, you've got 3 genes, they're actually doing different things, that somewhat overlapping, but there are differences. This is just tracking the number of gene conversions that you can track in this section of sequences. And as I said earlier, it's quite common, and this shows you that it's quite common. And the top, the blue, is giving you the maximum amount of recombination that you could have from a particular pattern of sequences, and the other plot is giving you the minimum. And here we're looking at the core alleles, and there are 42 of them. There's 11 HLA-As here, 17 Bs, and 14 Cs. And in a previous study, we analyzed a lot of the archaic human genomic sequences and found many of the modern sequences seem to have come from archaic humans, such as Neanderthals and Denisovans, and those are the ones that are all yellow. And you can see there's a lot of yellow there, and I think it's telling us the extent to which migrants coming out of Africa and going into Europe and Asia had to rely on supplementing their supply of HLA class 1 alleles with those that they could get from the resident archaic humans. We can also, from the numbers here, we can estimate the number of different alleles that we might expect to find in the human population. And the estimate is somewhere between 2 and 3 million A's, 2 and 3 million B's, and 2 and 3 million C's. And this is just showing that analysis. So this is the frequency. So when the bone marrow transplanters asked me, well, have we finished now? There's loads out there. And the thing is, with bone marrow transplantation, you're typing millions of people, of which only a very small fraction of them are ever going to be donors. And they're soon going to find some patient who has one of these very rare SNP alleles. And even though it's not useful in terms of responding to viruses, it could still provoke an alloreaction that would cause a graft versus host reaction in the patient. So that this acquisition of these alleles is probably going to continue for some time. But now we know essentially that you're going to get basically one novel allele in every 2,000 people that are studied. All right, now I'm going to turn more to sort of functional immunology and come back to this picture and just remind you that the way that natural killer cells work is that they get educated when they're growing up to be able to detect normal levels of HLA class 1 on cell surfaces. And then when they have become educated and circulate, they're able to respond to cells that have lost HLA class 1, which is a common sequelae of cancer. Many tumors downregulate class 1. Many viruses also downregulate host class 1. And so these are cells that are not healthy. And so normally inhibitory signals between HLA class 1 ligands and inhibitory receptors, such as CD94 NKG2A and also the KIR, are involved in inhibiting the NK cells. And then when class 1 is downregulated, you don't get that inhibitory signal, and then there's a balance in the cell between activating and inhibitory, and then the activating receptors go into the ascendancy and you get activation. And you see this, as I've said, in infection in cancer, but it also occurs in pregnancy when the maternal NK cells that are in the uterus and are involved in placentation are seeing a trophoblast cell that has paternal antigens that is looking as though it's not self. And so you get the same type of response. So these KIR, they don't detect all HLA class 1. They detect 4 epitopes, which are mutually exclusive, and they're part of the alpha-1 helix of the HLA class 1 molecule. And these are the different allotypes and the distribution of the epitopes. There's the BW4, which is found on both A and B, A3 and 11, which is only on some of the A's. You have, every C has either a C1 or a C2 epitope. And these are what the KIR recognized. And there are 2 different lineages of KIR. There's the lineage 2 KIR that interact with the HLA A and B epitopes. And then there's the lineage 3 KIR, which interact with the HLA C epitopes. And from what we know from the evolution and from studying pregnancy, where trophoblasts only express HLA C and not HLA A and B, there's a partition of work where I think that the HLA C is more involved in reproduction and that the A's and B's are more involved in the response to infection and to cancer. So I'm going to summarize an awful lot of work looking quite carefully at all these different higher primate species. But we can say that counterparts to human KIR and HLA class 1 ligands are really present only in the great apes and in the old world monkeys. And I'll just run you through the evidence for that, and we're just going to fill in the boxes as we go through this slide. So HLA-E, which is probably the oldest of the human HLA class 1 molecules, is found in both the new and old world monkeys, all the apes, and in humans, and so is the corresponding inhibitory receptor, CD94 NKG2A. In the old New World monkeys, there's nothing resembling HLA-A, but there is in old world monkeys and apes. They also have B, and they have BW4, which is that epitope that I showed you that interacts with the KIR that are interacting with HLA-AB. And the old world monkeys have lots and lots of genes encoding HLA-B equivalents that have these epitopes, and they have lots and lots of KIR of this lineage 2 that form the KIR that interacted with these epitopes. It's quite dramatic, but when you go on and you start to look at the orangutan, you find that that whole region of the locus that was full of these lineage 2 KIRs is gone. And there's basically one pseudogene left. And then the other part of the locus, which used to have one lineage 3 KIR, has now got half a dozen of these. And so between the separation of the orangs and the old world monkeys, they've gone off in different directions. And in orangs, you get Cs, which correspond to those KIRs, but they're all of the C1 type. So the C1 locus evolved, or the C1 epitope evolved before the C2 epitope. And we think it probably started off at a B locus because there are quite a few Bs in the chimp and a couple in humans that have retained the C1 epitope, which is not common amongst HLA-Bs. And so when you get to chimpanzees and humans, and also bonobos probably and gorilla, you have both the C1 and the C2. So from this analysis, we can see how the KIR locus and its epitopes have evolved and built up to complement the CD94-NKG2A interaction. And what I'm going to go on to show you is that as the KIRs have been brought into play, they've actually affected the older system of CD94 interacting with HLA-E. So just to review this, that the New World monkeys had KIR that are very different from human KIR. The Old World monkeys had numerous lineage 2 KIR that recognizes MHC A and B. There's a contraction of these in orangutans, and then you've got an expansion of the lineage 3 that recognize C1. The lineage 2 KIR are reduced to 1 gene in chimpanzees, and there's further increase of lineage 3 KIR, in the chimpanzee, and you have the evolution of both C1 and C2. The components are similar in humans, but the humans have a difference that's all of their own, which is that the KIR locus in humans has 2 very different forms. So you have haplotypes of the locus that are functionally quite different and complementary. And the chimpanzees don't have that. Okay, so let's go back. So let's have a look at the human KIR locus. So these are the A haplotypes, and these are the B haplotypes. And the A KIR haplotypes are more polymorphic in terms of their gene content. As you can see from looking at them, there's the haplotypes with different genes in different places. The A haplotypes are fairly constant, but the A haplotypes have greater polymorphism at the individual genes. And these numbers that I've put up are showing you the number of different alleles and then the number of different proteins that correspond to those alleles. And what you can see is that with some of these genes, we're getting sort of fairly substantial numbers of alleles. Here's 79 here, 26, 57 over here. And almost all of this work has been done in my lab. So for this KIR side of things, where the clinical applications have yet to be developed, we're not getting that type of effort yet. But just by working in my lab, we've been able to sort of put together a pretty substantial database. And so this is another system that is very, very polymorphic. And the peculiar thing about this system is that both the genes and both the ligands and the receptors are polymorphic. And so in a family where you've got ligands and receptors segregating independently, you'll have some children that will have a particular ligand-receptor interaction and others that won't. And so you've got to bring the two right things together. So there's maintaining a system that is inherently very unstable. So just from doing a lot of functional analysis, we can see that the KIR-A haplotypes have 2DL1 alleles, which encode strong inhibitory C2 receptors. But they have KIR-2DL3 alleles that encode weak inhibitory C1 receptors. And the B haplotypes are the other way around, that you have 2DL1s that are weak and C1 receptors that are strong. And to not go into great details, Ashley Moffat and her colleagues at Cambridge, down the road here, have shown that pregnant women who have a B haplotype are less likely to get preeclampsia, have low birth weight, have spontaneous abortion than women who have a B haplotype. And the worst combination is having 2 A haplotypes in the mother and a C2 ligand on the fetus. And there have been fairly extensive clinical correlations to show that Ashley and her colleagues have done. And so what we're seeing here is the balance that's being struck is between being able to combat infection and being able to survive epidemic disease, But then once you've got a decimated population that survived disease, then the competition is to who can replace the population. And so you get this type of balance that has been set. Many clinical correlations have been made with this KIR haplotype difference. The clinical labs are only doing an assay that measures the presence and absence of genes, so it's very low resolution and clear, but it's basically the same set of diseases that are correlated with HLA, except now we're correlating combinations of HLA and KIR. So these are autoimmune diseases, and these are infectious diseases, and there are also the reproductive diseases. We've worked with Ashley on a study to look at Ugandans. She's got huge collaborations with maternity hospitals in Uganda, and they have a very good cohort there. Well, there's a gene called KIR2DS5, which is an activating receptor for C2. Europeans have one allele for that gene, which doesn't work. Africans have 11 different alleles, of which 6 work and 5 don't work. And you see that the correlations with the reproductive conditions are different in Africans versus European, and that this 2DS5 gene, when it's a functional one, does provide protection against preeclampsia. So this also is emphasizing the need to study populations as populations, and that the human population has diversified tremendously as it's moved around the world. And this is another story that we have tried to investigate, but I haven't got time to talk about that here. So we'll just now sort of come back to some of what I've been saying, is that there are dual functions of HLA A, B, and C in NK cell biology. You've got polymorphic sequence motifs in the alpha-1 domain and of HLA A, B, and C that form the epitopes recognized by KIR. But you've got an indirect function, which is the anonomer peptide, which is cleaved from the leader peptide sequence of HLA A, B, and C polypeptides, binds HLA E, and is necessary for its surface expression and for interaction with CD94-NKG2A. So the polymorphic ones that are interacting with the KIR, with their mature form, are also influencing the immunology of this other receptor by providing the peptide that is needed to get the E to the surface where it can function. Now, the critical residue in the leader sequence is methionine at position -21, which anchors the peptide in the P2 pocket, as shown here. So this is B, C, E, and A, and here you cleave out this peptide, and then this is the peptide. This is methionine that interacts with the B pocket. All right, so if we look at the distribution of -21M, you get the following difference, is that all of the HLA-A alleles and all of the HLA-C alleles have that residue, so they can provide this binding peptide. Whereas HLA-B, which is the most polymorphic of the genes, -21M is the minority form, and -21T is the majority form, and the T form doesn't have this function. It doesn't stabilize the HLA-E, and it doesn't allow it to interact with CD94-NKG2A. So in the human population, this simple polymorphism, which is a SNP polymorphism, has the potential to diversify immune responses. And so I'll just go through how the effects of this polymorphism, because it basically is influencing the haplotypes that are carrying -21M at HLA-B and -21T. So this is what we call the old school of NK cell education, where you've got -21M, you get an HLA-B binding peptide, which can interact with HLA-E, and then you get good education of NK cells that carry the CD94-NKG2 receptor. To a lesser extent, you get some education of KIR through C1, but the C1 alleles that are on this haplotype are expressed at a lower level than the HLA-C forms that are on the other haplotype. So we call this the new school because you don't have an HLA-B binding peptide, but you can get your HLA-B binding peptide from HLA-A, so you can get some education of CD94 NKG2A. On this haplotype, you can have HLA-B and A that have the BW4 epitope, whereas the BW4 epitope is never found on this haplotype here. And so you can get education of BW4. The C1 alleles that are on this haplotype have high expression, and so they can get good education of KIR2DL23 NK cells. And you can also get education of C2 because you've also excluded C2 from the other haplotype. So in the evolution of going from an orangutan situation where you only have the C1 epitope to where you have both C1 and C2, that evolution has only taken place on a subset of these haplotypes. So we're sort of keeping the old form that we inherited from the orangutan, and then we're building on that by making another form. And so you clearly have a specialization between these two haplotypes, where the one on the top is clearly going for CD94 NKG2A, whereas the one on the bottom is really working a lot with the KIRs. So this simple polymorphism, one nucleotide change, is having a pretty strong influence on the way that the HLA locus is being conferred. And the prediction is that it will have an effect on the functional activity of NK cells. And so we all are either MM, MT, or TT. And so Amir Horowitz decided to do a fairly ambitious experiment to do an extensive immunological analysis of people who are MM, MT, and TT. And these are the other people who are involved. Because MM is relatively infrequent, we had to go to our Norwegian colleagues, where there's a lot of the haplotypes that have those particular alleles. So there's a bias towards Norwegians in this sample. So the -21M increases HLA expression. That's what we expect. That makes the binding peptide. It will allow HLA-E molecules to come to the surface where they can interact with receptors. And you can see that here. This is looking at each of these cohorts of 20, that if you have MM, you have more HLA-E on the surface. MT is not quite as good, but TT is much lower. And in every assay that I'm going to show you, we have this same pattern where it's just the presence of M is enough. Having 2 copies of M doesn't give you a whole lot more. M is dominant over TT. -21M reduces the frequency of these NK cells, as you can see here. So if you have MM, you have fewer. And this is a characteristic of good education. Like in a good university, you take in a lot of students and only a few of them come out. And so this is characteristic of educated NK cells. And you also find that it reduces the NK cell expression of the CD94 NKG2A receptor. And this, again, is a characteristic phenotypically of the educated NK cells. So this is showing that this polymorphism, where you have the M, you're getting better education of your CD94 NKG2A NK cells. This is an assay to look at what's called ADCC, which is the type of biology that's going on when the patient is treated with rituximab as a therapeutic antibody against B-cell lymphoma. It binds to the tumor cells, and then the NK cells have an FC receptor that binds to the antibody and kills the tumor. And you see that the people who are MM have a better reactivity in this assay. Here's another assay which is looking at the ability of NK cells to make inflammatory cytokines, such as IL-12 and IL-15. And again, the MM individuals are making stronger responses. This is another analysis which is looking at the number of NK cells expressing CD94 NKG2A using CyTOF. And I would just point you to the last column. And you can see that if you're TT, you have less diversity of your NK cells than if you're MM. And this, again, is a reflection of the better education with the MM. So by these functional criteria, you're seeing some quite dramatic differences between these three different phenotypes. And I'm sure that everybody who has a disease cohort is taking a simple PCR assay and can go and look through all their patients and see if you can get correlations with this polymorphism. It has been correlated with responses to AIDS already. And what I would offer up to you is that when you do these very, very complicated analyses, you can sometimes come out with very simple things at the end, which could then have some clinical application. So the KIR haplotypes, and this is more of a summary, is that the KIR A's give you stronger C2-mediated education, weaker C1-mediated education. KIR B haplotypes give weaker C2-mediated education and stronger C1-mediated education. -21M haplotypes, stronger NKG2A-mediated education and weaker KIR-mediated education. And the -21T, it's weaker NKG2A and stronger KIR. And I didn't show you that data. So this is an example where the KIR have led and where the HLA has followed up. Most of the time, it's the other way around. The HLA is leading and the KIR are responding to that. But then we're getting the other thing happening, that you get this polymorphism, and then it changes the way that the HLA have evolved to catch up with the KIR. So in summary, conserved and polymorphic MHC class I molecules are ligands for receptors that enable lymphocytes to detect and respond to cellular changes in MHC class I expression. In humans, the interaction of HLA-E with CD94 NKG2A is a conserved interaction that is shared with apes and old world monkeys. This interaction is, however, modulated by a human-specific leader peptide polymorphism of HLA-B, and as a consequence, HLA haplotypes divide into two groups that differentially educate NK cells. And KIR are highly polymorphic receptors that interact with highly polymorphic HLA-A, B, and C ligands in ways that are considerably species-specific. Thus, only 3 of the 14 human KIR genes have chimpanzee orthologs. That is our closest relatives we only share a few of the KIR. Distinguishing the human KIR locus are 2 forms of KIR haplotype that are functionally differentiated and have different associations with a spectrum of disease. And defining these differences requires combining population genetics and population biology with functional immunology. And I just like to finish off with a little vignette that about 15 years ago I was invited to go on a site visit to ILRAD, which was then the animal research center in Kenya, and it was to deal with MHC. And the first thing they asked me, I said, well, and they were studying various species, I said, you've got to do the population genetics and study the MHC in all these animals. And they just laughed at me. And they said, there's no way we're ever going to do that. There is now the way to do that, much more cheaply and much more effectively. But if you don't dig into the population genetics and biology, you're just acting in a way that is with the best one eye open. And so that would be my recommendation. So thank you very much. It's been very nice talking to you.
So you mentioned at the end, towards the end, that the haplotype is very rare. And is it very rare? I mean, is it rare because it has a disadvantage, do you think?
Well, when I say rare, it's probably at about 25-30% frequency in European. So it's not very rare. It's a minority. And I don't think that there's so far that there hasn't been a disease or a condition that's been associated with it, but this hasn't been looked at. I mean, our paper on this only came out in December of last year, and before that, it had, as a possible disease marker, this polymorphism had been basically ignored, and there was one paragraph in a very obscure journal of an article by an HLA typing expert called Edmund Eunice, who picked up on this. And the context in which he was writing about it was that he noticed that all of the haplotypes that have BW4 have minus 21T. And in the AIDS area, they had been correlating all sorts of AIDS factors with BW4. And the fact is that when you have a disease association with BW4, you don't know if it's BW4 or it's minus 21T. And so this is very new. And I'm sure we're going to see a lot of analysis in the near future.
Thanks, Peter. Actually, related to that, maybe I haven't understood correctly, but so these populations of cells with the M heterozygote and TT, were they from Norwegian? Were they all Norwegian? Or?
No, they weren't all Norwegian.
So, so the question I've got is, how do you you're looking at that allele, but how do you control for the underlying genetics and potential stratification in a study like that?
We don't. I mean, it's. We're working at Stanford. We go to our blood bank and ask for 25 units of blood. We go to lots of Chinese people and Vietnamese people, and we don't get many classic. And we needed homozygotes as well. We get the heterozygotes, but no, we aren't. There's so many underlying things that
could be there.
It could be, yeah. But these are the haplotypes that are the ones that have been associated with all those autoimmune diseases. MS, diabetes, classic A1B8, DR3 is haplotype. And these are also haplotypes that we think come from Neanderthal. So I'm sure there are benefits and there are detriments, but I think that is, with all of these factors, they're all balancing one thing off against another.
Or they wouldn't be there.
Yeah. I mean, some of the best studies that one can do are looking at these ethnically well-defined small population groups, because in those groups you have small numbers, relatively small numbers of HLA and KIR, and you can see how ones have been pushed up to very high frequency. So, for example, there's a population living on a mountain between Venezuela and Colombia called the Yucpa, who are about 1,000 people who've been through measles and other epidemics in the recent past. And they have pushed up to very high frequency a B allele called B39, which is relatively uncommon in Europe, but that is an M allele, and it's at the highest frequency in the world in that population. And so the evidence that has actually been doing something, I think, is it really reinforces that.
So here?
Yes.
So you said that several of the HLA alleles were from archaic genomes. So does that mean that you don't find those allele types in Africans?
Yes.
Okay.
Yeah, that's one of the criteria.
Yeah. But it looked like there was a quite large proportion of diversity.
It does.
So are there more diversity in Europeans and Asians than in Africans?
No, because humans have been living in Africa for hundreds of thousands of years more. So there are a lot of African-specific alleles.
Yeah.
But the comparison between Africans and Europeans is also very valuable because in terms of haplotypes, they're quite different.
So is it more than you expect by chance? We know that there's been integration between archaic humans and modern humans. So had there been a higher proportional contribution at this? Because you were indicating that this was part of adaptation. Because then you should see there should be enrichment compared what you expect by chance. Did you see that?
Well, I don't think we know what chance is because we don't know what the sizes of the numbers of people that were.
Yeah.
I mean, there are many debates. One is that there wasn't much intermingling.
Yeah.
And that if that's the case, then when they have acquired archaic haplotypes, it's probably more likely to have been subject to selection than if they are intermingling in the same way that in California, people from Asia are intermingling with people from Europe.
I have another question also of mine. So you in your abstract you say that mouse doesn't have gears.
Yes.
So when did they arose and where did they come from, so to speak?
Yeah. So we've done a sort of a genome project mining, and you find that gears are only found in higher primates. They're not in lemurs and prosimians, and they're also present in
all ruminants. Okay.
all ruminants.
Yeah. I'll show you later.
Yeah. And the mouse doesn't have gears, but it has a family of genes called LY49 that are lectin-like receptors that encode receptors that interact with polymorphic determinants of the mouse class 1. And an extraordinary number of their properties are the same of the KIR, but they have clearly evolved independently. But they must have been under such similar selection pressures. And they use all the same signaling circuits inside the cell. And when another group did a data mining with the LY49s, they found that they're only in some rodents and they're in equids, donkeys, and zebras. And there's a hint of data, hasn't been followed up, that the prosimians are using the CD94 NKG2A, which they have diversified for the same.
Yes, they've been lost rather than that the common ancestor all had multiple, and then certain lines have been lost, so to speak, during the.
Yeah, and we have examples which we can see in the primates. The gibbons, it looks as though their KIR locus has been hit by a bomb. The genes are all in bits, and it doesn't look as though it works. And it's happened in at least 4 animals from 2 different species. And we can see in humans that what's happened with the B haplotypes is that there's been gradual attenuation of some of the genes. And on the A haplotypes, it's gradual attenuation of some of the genes. So that it is a very dynamic locus. And the issue that I raised before is that in every family where you're mixing genotypes, you haven't got stable sets of ligand receptor interactions. So this system is probably susceptible to losing its way, and where you have maybe a small population that's only got the ligands and doesn't have the receptors and so on and so forth. And under those circumstances, it would then decay.
I'm curious to hear that HLA has been driving care evolution, which is sort of a recent finding, I would suggest. Is that because the polymorphism in the leader has no impact on TCR function? So if there's no dual role for that polymorphism in the other arm of what?
No, HLA does interact with. There are some T cells that interact with, and it doesn't have to be absolute. It's just whichever is in an ascendancy at a particular time. But I think that the HLA class 1s have been far more stable in general than the KIRs. And so it looks as though that they're the thing that doesn't get lost. But you clearly can lose care and survive as well.
Okay, well, can I thank you again, Peter?
Thank you.